clinical pharmacology studies to support filing packages and labeling. Since the overall objective of my talk today is to introduce and explain about the clinical pharmacology studies that are critical to inform drug labeling and filing packages, I will briefly talk about the significance of these studies in general on the overall drug development. Then I'll introduce each of these ClinPharm studies and discuss various aspects such as the study conduct, the information obtained from these studies, and how this information can support drug labeling and filing packages. Okay, so what is clinical pharmacology and its significance of these studies in the drug development? Clinical pharmacology studies aim to understand the safety and tolerability profile of the drug in humans across a range of doses, ultimately to determine the appropriate dose for the intended population. Clinical pharmacology studies also aim to evaluate um, and understand the pharmacokinetics of the drug by studying the drug's absorption, distribution, metabolism, and excretion properties. ClinPharm studies also aim to understand the pharmacodynamics or the PD of the drug by aiming to understand both the desired effects of the drug and the unexpected effects of the drug. These ClinPharm studies are usually carried out in healthy subjects, typically including both males and females, since this allows testing of the PK, PD, and safety profile of the drug without the influence of any pathological conditions. In some cases, like for example in oncology drugs or for specific population, these studies may not be conducted in healthy volunteers. The clinical pharmacology studies can benefit the drug development process by providing a better understanding about the PK and PD of the drug and thereby help optimize the dosing regimens and the formulation. These ClinPharm studies also help evaluate the appropriateness of administering the drug with and without food by studying the food and drug interactions. These studies could also help mitigate the risk to patients by better understanding any possible drug-drug interactions. These ClinPharm studies also provide a better understanding of the safety and efficacy profile of the drug in specific populations, such as renally impaired or hepatically impaired patients. In addition to this, these studies also help in defining the inclusion and exclusion criteria for the late phase studies. Due to all of this information that is obtained from these clinical pharmacology studies, these form an integral role in the health authority submissions and inform the prescribing information. Now, in order to understand the impact of ClinPharm studies on the drug label, let us have a look at the table of contents of a drug label. The sections highlighted here with the boxes in red represent the sections in the drug label which are informed by the data obtained from these clinical pharmacology studies. As we proceed in the presentation today, we will see what information is obtained from each of these clinical pharmacology studies to inform these highlighted sections in the label. And to understand the impact of these clinical pharmacology studies on the filing packages, here is a picture. Here is a picture of the Electronic Common Technical Document Triangle, or the ECTD triangle. The information that is provided to the FDA for approval of the drug is recommended to be submitted in the ECTD format. The ECTD contains an electronic table of contents that is broken down into 5 modules. These different modules are represented in this ECTD triangle shown here, of which the module 2.5, which is the clinical overview, and the module 2.7, which is the clinical summary, are the sections which are informed by the clinical pharmacology studies. Now, these are the different clinical pharmacology studies which are listed on the slide here. These are conducted for an investigational drug. Now, which of these studies are needed to be conducted depends upon different factors, such as the disease area, the modality of the investigational agent, the route of administration, or the stage of development. For example, if the drug is being developed for a disease area in which the patients with hepatic impairment are not eligible for the treatment, then the studies for that investigational drug might not be required in the hepatically impaired patients, even though the drug is known to undergo hepatic metabolism. Also, with respect to the drug modality, for small molecules, we need to conduct almost all of these ClinPharm studies listed here to understand the behavior of the drug in vivo. However, a lot of these ClinPharm studies might not apply to the therapeutic protein or biologic. Now, in case of route of administrations, some studies such as the food drug interactions or the drug interactions with the acid reducing agents could be required for the drugs which are orally administered. But if we take the same drug and make an IV formulation out of that, then these studies might not be required to be conducted. So we really need to understand the characterization of the drug to design these clinical pharmacology strategies for the development of a given drug molecule. Now, in the earlier slide, we saw which of these clinical pharmacology studies are to be conducted. Now, let us look at a figure to understand when these studies need to be conducted during the drug development process. Now, most of you must have seen the schematic of different stages involved in the clinical development. After the data from the preclinical studies is collected and an initial IND is submitted, then we begin our clinical pharmacology studies. The earlier you can get the information from these ClinPharm studies, the better it is, since in that way we can help inform the late phase studies, and this helps maximize the chances of success for the drug. Sometimes an early food effect study or early food effect assessment may be conducted during the first in human studies before conducting a complete food effect study. Also, a mass balance study may be done early on in the drug development to understand if the route of elimination for the drug is renal or hepatic, and depending on that, either a hepatic impairment or a renal impairment study might be required for that particular drug. However, the decision of when and how to do these studies depends on the drug product, and this needs to be carefully considered for each and during the clinical development. Now, let us look at the very first studies conducted in the humans, which are called as first in human studies. Since the drug is tested in humans for the first time in these studies, these are considered as bridges to the future clinical development. The objective of these FI studies are to generate the initial knowledge on the safety, tolerability of the drug, and the PKPD of the drug. These studies allow to evaluate the single doses in case of single ascending dose studies and steady state pharmacokinetic parameters in case of multiple ascending dose studies. These FI studies also allow the evaluation of some exploratory objectives such as the food drug interactions, or they also help in identifying metabolites or biomarker response in some instances. These first in human studies are usually randomized placebo controlled studies and are typically conducted in about 20 to 80 healthy volunteers. Again, this might not be the case for oncology drugs. Determining the starting dose for these FI studies that has an acceptable safety margin is critical prior to initiating these FI studies. Now, the starting dose selected for these studies should be such that it should balance the need to elicit any pharmacological response, at the same time mitigate the risk of toxicity. Typically, the starting dose in these FI studies is determined by the available non-clinical pharmacology and toxicology studies, as well as any available human experience with any other therapeutic areas is used to determine the starting dose. Now, there is no singular approach to selecting a proper FI starting dose due to the uniqueness of each new drug candidate, and the regulatory guidances also recommend various approaches to determining the starting dose. The most widely used approach follows the FDA guidance for estimating the safe starting dose by converting the no observed adverse effect level, or the no AEL, obtained from the most sensitive tox species to the human equivalent dose with the use of allometric scaling. For certain classes of the drugs, toxicity may arise from exaggerated pharmacological effects, and in this case, a pharmacologically active dose or a PAD may be more sensitive indicator of potential toxicity than the no AEL and may warrant lowering of the uh, recommended starting dose. Or in some cases, a minimum anticipated biological effect level, or a MABEL, which is based on pharmacology rather than empirical scaling factor, might be the safest approach and which can be used for the high risk drug candidates. Now, there are several study design considerations which need to be considered for conducting these first in human studies. Typically, these FI studies enroll about 8 to 10 subjects per cohort, and the subjects are randomized as 3:1 or 4:1 ratio. Most of these FID study employ a dose escalation design, and there are two basic approaches of dose escalation. It could be either a rule-based approach or a model-based approach. In case of multiple ascending dose studies, the duration of dosing depends upon the half-life of the drug in order to allow for adequate time to reach the steady state. Since these studies are testing the drug in the humans for the very first time, these studies require a safety monitoring plan and risk mitigation measures in place for the safe and ethical conduct of these trials. Traditionally, single ascending dose studies with a separate staggered multiple ascending dose studies in parallel were conducted, but combining these single ascending dose studies and multiple ascending dose studies significantly shortens the timelines compared to conducting separate single ascending dose and multiple ascending dose trials. These initial trial studies also provide an opportunity to assess the food effect or drug-drug interaction potential or the formulation effect early in the drug development, and these can be included in the single ascending arm of the study when the dose escalation has reached an anticipated therapeutically relevant level. So what are we looking to gain from these first-in-human studies that would inform the drug label and the regulatory filing? These studies provide some initial safety and tolerability data, possibly a maximally tolerated dose. These studies also help generate general pharmacokinetic characteristics of the drug, including the variability, PK linearity, and dose proportionality. These studies also help to obtain the steady state parameters from the multiple ascending dose studies. Now, we will look at some of the clinical pharmacology studies which are required early in the drug development for an oral formulation. And these studies include the bioavailability assessment, the food effect assessment, and the pH effect assessment. There are FDA guidances for each of these studies which talk in detail about how these studies are to be conducted, what data should be collected for these studies. But for today's presentation, I'll touch upon a few of these key points from each of these studies. Now, as I have mentioned earlier, most of these studies are typically conducted in healthy adult participants. Very rarely, these studies are conducted in patient population due to safety concerns or if differential effects are expected due to underlying disease condition. Now, before looking at the first bioavailability assessment and the study considerations for the bioavailability study, let's do a quick recap about what is bioavailability. Bioavailability of a drug is defined as the rate and extent to which the drug is absorbed from a given formulation and becomes available at the site of action. These bioavailability studies provide pharmacokinetic information regarding the relative fraction of the drug that is absorbed into the systemic circulation. These studies also provide dose proportionality or PK linearity information. Now, for new drug applications, these BA assessments are usually carried out at the pre-approval stage. Sometimes they may also be necessary at the post-approval stage. During the pre-approval stage for the NDA, these bioavailability assessments are conducted to compare the early and the late phase clinical trial formulations. These may also compare the to be marketed formulation with the clinical trial formulation in case these are different. These studies also compare the equivalence of the product strength. For example, they compare the dosage strength proportionality. For example, the dosage strength proportionality study comparing the extended release formulation to the immediate release formulation. And these studies could also compare the new and the previously approved product in support of a 505(b)(2) application. Sometimes these bioavailability studies are also conducted during the post-approval stage, and they are required to bridge the post-change product to the pre-change product. These could support these supplemental applications when there is a major change in the manufacturing site or manufacturing method, etc. Now let's look at the general considerations for the bioavailability studies. The objective for conducting these bioavailability assessment is to understand the rate and extent of the drug absorption from the test formulation relative to the reference formulation. Typical bioavailability studies are conducted as a crossover studies, and sometimes parallel design studies might also be conducted depending upon the PK characteristics of the drug. The crossover studies definitely reduce the variability in the PK measures that may be caused due to subject-specific factors and thereby increase the ability to discern the differences in the PK measures caused by the formulation itself. However, for the drugs with long half-life, a parallel study design might be used where a crossover study might be impractical. Now, here is a schematic for a typical bioavailability study, which shows a typical crossover bioavailability study. Now, it is essential to have an adequate washout period between the test and the reference formulation arms in order to ensure that there are no carryover effects. Now, the FDA guidance recommends conducting a pilot bioavailability study with a small number of subjects under certain circumstances, and the main purpose is to gain the experience before conducting a pivotal bioavailability study, mainly to optimize the sample collection time intervals or to determine the length of the washout period. Typically, these BA studies are single dose studies conducted under fasting conditions since these are more sensitive, but there are some scenarios under which multiple dose studies may be warranted. If multiple dose studies are conducted, then the drug is to be administered till the steady state is reached. Also, if there are any tolerability issues under the fasted conditions, then the drug may be administered with the food. Now, these bioavailability studies are interpreted by applying a confidence interval criteria where the test and the reference formulation would be considered similar if the 90% confidence interval of the exposure measures is within the bioequivalence limit of 80 to 125%. In addition, for highly variable drugs, the reference scale average PE approach or the RSABE approach may be utilized. This method allows to scale the acceptance bioequivalence window based on the within subject variability of the reference drug, thereby permitting to increase this bioequivalence window as the variability increases. Also, it is important to note that when similarity in the bioavailability is not demonstrated in these bioavailability assessment, then it is essential to demonstrate that the rate and extent of the absorption do not meaningfully affect the safety and efficacy of the drug based upon the exposure response analysis. As we have seen earlier, these BA studies provide important pharmacokinetic information related to the dosage regimens, which will ultimately support the clinical pharmacology sections of the product label by providing information on the relative fraction of the dose absorbed or any linearity, non-linearity in the pharmacokinetics. The results from these bioavailability study will also help guide the dosage and administration section in the label. With respect to the filing packages submitted to the regulatory authorities, these BA studies provide information regarding the drug exposure of the test formulation. And in some instances, these BA studies also help support or bridge the approval of a formulation with a novel formulation. And one such example of this bridging was shown for tofacitinib extended release approval, which I'll be discussing on the next slide here. Now, in this particular case study, a relative BA study was conducted post the approval of tofacitinib immediate release formulation for bridging the bioavailability of the novel extended release formulation. Now, tofacitinib is an oral Janus kinase inhibitor, which was approved for the treatment of autoimmune disorders, including rheumatoid arthritis. The first approval for rheumatoid arthritis was for the immediate release formulation, which was to be dosed twice daily, and the label was based on this immediate release formulation. Later, the sponsor developed an extended release formulation, which required once daily dosing. Now, the approval of the extended release formulation was based only on the relative bioavailability studies conducted, which demonstrated the bioequivalence between the extended release formulation once daily dose and the immediate release formulations twice daily dose, and no additional safety and efficacy studies were conducted for the approval of these extended release formulation. I'm sorry. Now, moving on to the next ClinPharm study, which is the food effect assessment. Now, the presence of the food in the GI tract can interfere with the absorption of the orally absorbed drugs, and this impact on the exposure can affect the safety and efficacy of the drug products. These effects can be manifested in many different ways. Now, the intake of the food may either increase the gastric pH, thereby interfering with the solubility of the drug. It may also delay the gastric emptying time and thereby increasing the resident time of the drug in the stomach. Food may also increase the bile salts that may sequester the study drugs. They could also alter the first pass metabolism, or they also interact with the uptake or efflux transporters. And hence it is important to assess the effect of food on the orally absorbed drug products early in the clinical development, as this will not only help optimize the safety and efficacy of the drug, but it will also inform the dosing recommendations for the late phase studies, as well as provide adequate instructions for the administration in relation to food in the product label. Now, the main objective of conducting a food effect study is to evaluate the presence of the food um, on the rate and extent of the drug absorption from the given formulation. Now, higher exposures under fed conditions might increase the risk of clinically significant adverse events, and some drugs which are known to cause GI irritation can be administered with food to elevate this irritation. Now, these food effect studies are typically randomized studies, single dose studies. Usually, the clinically recommended dose of the drug is used, or in case of multiple doses, or sometimes multiple doses which exhibit linear PK are to be marketed, then the highest therapeutic dose would be used for these studies. These could be two-way or three-way crossover studies depending on how many meals are evaluated or how many formulations are evaluated in these studies. This could enroll both male and female participants, and these studies required adequate washout period between the two treatment periods. The guidance does not specifically recommend the number of subjects for the food effect studies, since it will depend upon the PK variability of the drug. Now, differences in the fat content or the composition of the meal can have a varying impact on the magnitude of the food effect. And hence, the FDA guidance recommends that these food effect studies are typically conducted with a high fat meal, since these represent the worst case scenario and are most likely to affect the GI physiology. The food effect study with a low fat meal should be considered if there are significant impact with the high fat meal, and since the low fat meal can have less or no impact on the systemic exposures of the drug. The food effect study can be conducted early in the drug development, and these are typically the pilot food effect studies. These help with providing a preliminary food effect assessment, which may be important for assessing the dosing requirements for the subsequent late phase trials. Now, the definitive food effect studies are done to assess the food effect for the formulation used in the pivotal safety and efficacy studies, and to determine the dosing instructions for the product label for the to be marketed product formulation. Now let us look at the interpretation and impact of these food effect studies. The exposure measures and the PK parameters should be derived and reported from these food effect studies, and these are similar to what is reported for the bioavailability studies which we saw earlier. Now, if the 90% confidence interval for the fed and fasted ratios fall within the 80 to 125% bounds, then it could be concluded that there is no food effect. However, if the effect of the food is observed, then the observed increase or decrease in the systemic exposure of the drugs in presence of the food might not be clinically relevant based on the exposure response information. And hence, it is critical to understand the exposure response relationship of the drug to assess the implications of these food effect studies. The food effect studies support the labeling recommendations pertaining to effect of the food on the PK and PD of the drug in the following sections in the label. It informs the dosage and administration sections, which could include specific dosing instructions relating to the food, whether the drug is to be taken on an empty stomach or it is to be administered with food or without regards to meal. These studies could also inform the clinical pharmacology section in the label by providing informing the effect of food section on the absorption of the orally administered drugs under the specific pharmacokinetic subsection. Other sections like the warnings and precaution sections, patient counseling sections would include appropriate information regarding to administration of the drugs with regards to the food. Now here is a case study of venetoclax for which the food effect study was able to inform the labeling recommendations for this particular drug. Venetoclax is a BCL2 inhibitor which is approved for the treatment of hematological malignancies. Now here on the left hand side is a snapshot from the drug label for venetoclax, and we can see that under the pharmacokinetic section where the food effect study for this drug was done with both low fat meal and high fat meal, and it was found that in presence of a low fat meal, there was increase in the venetoclax exposure by approximately 3.4 fold, whereas the administration of the high fat meal increased the exposure by about 5-fold of the drug. However, when we look at the dosage and administration section in the label, it is recommended that the patients take venetoclax tablets with the meal and water. And if you notice that they have not specified if it is to be taken with low fat meal or a high fat meal. And this was because of the exposure response analysis which was conducted, which showed that venetoclax could be administered without any specific recommendations for the fat content of the meal to ensure adequate and consistent bioavailability. Now, the next clinical pharmacology study that we will see is the effect of pH effect assessment to support drug labeling. Now, it is essential for the oral formulation that the effect of pH would be evaluated, since acid reducing agents, which are commonly used for the treatment of certain gastric disorders, do cause elevation of the gastric pH, which in turn can affect the solubility and dissolution of some of the orally administered drugs. As a result, the concomitant administration of an investigational drug with these acid reducing agents could alter the bioavailability of the drugs, and hence it is important to assess the susceptibility of an investigational drug to these pH dependent drug drug interactions. Now, a drug is said to potentially have a drug drug interaction with an acid reducing agent if the average Cmax or AUC is decreased by greater than 25% in the presence of an acid reducing agent. However, again, the clinical significance of this change is to be determined by the exposure response relationship. Now, typically, these studies are usually crossover studies since they reduce the between subject variability. And for long half-life drugs, a parallel study design could be considered. Now, on the right-hand side here is a framework to assess the clinical DDI risk for the drugs with acid-reducing agents. Now, since most of the drugs that demonstrate pH-dependent drug-drug interactions are weak bases, this framework here is for immediate release products for weak bases. Now, the first question to ask here is, does the drug have a pH-dependent solubility at the physiologic pH? If the answer to that question is yes, then we need to know if the drug is the drug solubility low at pH 6.0 to 6.8 compared to the solubility that is needed to dissolve the clinical dose. That is the therapeutic dose divided by 250 ml. If the drug indeed has a low solubility at pH 6 to 6.8, then a clinical DDI study with acid reducing agent might be required. Alternatively, there is an option to compare the dissolution and the different media conditions. And if it is found that the similarity factor or the F2 value is less than 50%, then a clinical DDI study is recommended. Now, for conducting these clinical DDI study with acid reducing agents, the choice of the acid reducing agents and the dosing regimen depends upon the purpose of the study, whether it is to characterize the worst case scenario or to identify any mitigation strategies. And the different choices of the acid reducing agents that could be used for these studies are proton pump inhibitors, H2 blockers, or antacids. Now, proton pump inhibitors irreversibly bind and inactivate the proton pump, leading to long-term suppression of the gastric acid production. If PPIs are used for the pH assessment studies, then it is required that a pretreatment for at least 4 to 7 days is required to achieve a pharmacodynamic steady state before administering the investigational drug. In case of H2 blockers, these are needed to be administered only a couple of hours before the administration of an investigational drug, and this is sufficient to elevate the pH. However, these H2 blockers are known to have relatively shorter duration of action. In case of antacids, they directly neutralize the gastric acid and they provide a quick onset of action, and they do have a very and these are very short acting acid reducing agents, and hence a concomitant administration of antacids with the investigational drug could be used for conducting these studies. Also, since the pH dependent drug drug interactions can be formulation dependent, it is important to conduct these assessments with a to be marketed formulation. Now, in case of the dose for the investigational drug, to reflect the worst case scenario, the highest intended therapeutic dose should be used for the investigational drug. For the acid reducing agents, the highest dose that is commonly used in the clinical practice is usually used for these studies. Now, these studies could be either conducted in fed or fasted state, depending upon the intended administration of the investigational drug. Now, generally, the clinical results from these DDI studies with acid reducing agents can be extrapolated to other acid reducing agents within the same class. In order to support the labeling recommendations, the FDA guidance provides a framework to extrapolate these DDI study results. Usually, the study with a proton pump inhibitor, since these are long acting, they represent the worst case scenario. Now, whether these changes are considered clinically significant or not depends upon the exposure response relationship. Now, if there is a clinically relevant drug-drug interaction effect observed with a proton pump inhibitor, then there are several implications for the labeling recommendations, and these are as follows. A clinically relevant DDI with PPI obviously means that these PPIs would not be used along with that investigational drug. Alternatively, the sponsors could consider conducting further studies to evaluate if the DDI still exists for the lower doses of these proton pump inhibitors. In case of H2 blockers, it is also recommended to avoid these H2 blockers. The sponsor could further conduct studies to evaluate if any staggered dosing with H2 blockers could mitigate these pH-dependent drug-drug interactions. And for antacids, because of their short actions, it is a staggered dosing is recommended and could be used even if a clinically relevant DDI is observed with proton pump inhibitors. Now, depending on the results from these studies, the following labeling sections would be informed. The clinical pharmacology sections, the warnings and precautions sections, the contraindication sections in the label. There would be boxed warning in case these lead to any serious drug-drug interactions, and the patient counseling section would also involve any instructions specific to the administration of the acid-reducing agents with the investigational drug. Now let us look at the studies which are conducted to assess the ADME properties of the investigational drug. And these are drug-drug interaction studies, mass balance studies, and renal and hepatic impairment assessment. Now, one of the important aspects of drug development is the assessment of the potential of drug-drug interaction with the investigational drug. These drug-drug interactions could occur when the concomitantly administered drugs interact with the drug metabolizing enzymes or the drug transporters, either by inhibiting or inducing their expression, and thereby resulting in alteration of the drug exposure. These drug-drug interactions could result in increase in the exposure of the investigational drug, which might potentially affect the safety profile of the drug, or there could be a decreased exposure of the drug, which might lead to reduced efficacy. Now, some of the most common drug metabolizing enzymes are cytochrome P450s and UGTs, and some of the most common transporters are PCRP, OATPs, and PGPs. Now, these clinical DDI studies could provide valuable information to inform the various sections of the drug label, such as the drug-drug interaction sections, which would obviously include the results from these drug-drug interaction studies. The boxed warning sections would contain any serious warnings or any contraindications. The dosage and administration section would include any information about any modifications in the dosage that is needed due to these significant drug-drug interactions. The contraindication sections, the warnings and precaution sections would include any additional patient, would include if any additional patient monitoring is required or if any dose adjustments are required, or these might also include any mitigation strategies. And obviously the ClinPharm section would include the changes that are seen based on these drug-drug interaction studies. Here are two FDA guidances relating to the conduct of these drug-drug interaction studies. One is for the in vitro DDI evaluations, and the other is for the clinical DDI studies detailing the CYP-mediated and transporter-mediated interactions. Now, let us look at how these drug-drug interaction assessments are conducted. Now, during the drug-drug interaction assessments, the investigational drug could either be the drug that is impacted by these interactions, that is The exposure of the investigational drug itself changes due to induction or inhibition of the drug metabolizing enzymes and transporters. That is, the drug itself is a victim of these drug interactions, or it might, or the investigational drug itself might alter the pharmacokinetics of the co-administered drug. This means it could be the perpetrator of these drug-drug interactions. So first, we start with in vitro test or screening to determine if the investigational drug is a substrate or inhibitor or inducer of these common drug metabolizing enzymes and transporters. If any of these initial tests are positive, then there could be a potential for the drug-drug interaction and a clinical DDI study is recommended. Now, after conducting the clinical DDI study, and if there is a well-established in vitro to clinical correlation, then a PBPK approach could also be used to test DDI potential for different scenarios, such as alternate doses could be tested, or alternate dosing regimens or formulations, or different candidate conmit scenarios could also be tested. Now let us look at a scenario where the investigational drug is a victim of these drug-drug interactions. That is, it is a substrate for the drug metabolizing enzymes or transporters. So the goal in such case is to see how modulating the enzyme or the transporter could impact the exposure of the investigational drug. To conduct the studies, it is recommended to use the investigational drug at a clinical dose, or if the drug exhibits dose proportional PK, then any lower dose in the range where the exposures to the drug increase in a dose proportional manner could be used. Usually, the single dose administration of the substrate is acceptable. For these studies, index perpetrators, that is index inhibitors or inducers, are used, and these are usually administered to steady state to maximize the potential of the drug-drug interactions. Now, the FDA website lists these index inhibitors or inducers. Now, here is a general schematic of such a clinical DDI study. The investigational drug is usually administered on day one after the subjects are screened into the study. The PK is collected, and after sufficient washout period, the perpetrator, which could be an inhibitor or inducer of enzyme or transporter, is administered to steady state. And once the steady state has reached, the single dose of the investigational drug is administered again, and the PK of the investigational drug alone and in presence of the perpetrator is collected, and the ratio of the exposure measures is determined. Now, the results of the DDI study are interpreted based on the no effect boundaries for the substrate drug. The no effect boundaries represent the interval within which a change in the systemic exposure measures is considered not significant to warrant any clinical action. Now, to do this, the ratio of the exposure measures, that is the Cmax and AUC, and the 90% confidence interval with and without the perpetrator is calculated, and um, and these can be interpreted using the following two approaches. The first is a conservative approach, which is to use a no effect boundary of 80% to 125%. And when the 90% confidence interval of the exposure ratios fall within the 80 to 125%, then it could be concluded that there is no clinically significant DDI. These 80 to 125% boundaries represent a conservative standard for the drugs that have wide safety margin and might not be ideal for using with the drugs with wide therapeutic index. Another approach which is a preferred approach is based on the no effect boundaries, which are developed based upon the exposure response relationship. Now let us look at a scenario where the investigational drug is a perpetrator of drug-drug interaction. That is, the drug modulates the expression of the drug metabolizing enzymes or transporters, affecting the exposure of the concomitantly administered drug. So the goal in such case is to see how modulating the enzyme or the transporter by the investigational drug would impact the exposure of other medications. Now, since the investigational drug is a perpetrator, it is administered to steady state. A prototypical substrate is administered at a single dose, and the dose of the investigational drug used is usually the maximum dose that would be evaluated in the phase 2 study. Again, this is a schematic similar to what we had seen earlier, where the perpetrator is administered to steady state, and the PK of the substrate with and without the perpetrator is collected. Similar to the previous study, the results are interpreted based upon the no effect boundaries. The ratios of the geometric mean of the exposures and the 90% confidence interval are calculated with and without the presence of the inhibitor or inducer. Now, the investigational drug could be classified as either strong, moderate, or weak inhibitor or inducer based upon the change in the AUC of the substrate, and this classification is provided in the FDA guidance. Now, another approach to conducting the DDI study is doing a cocktail DDI approach. The cocktail DDI study includes simultaneous administration of the substrates of multiple CYP enzymes or transporters to the study subjects. Now, the advantage of using this approach is that it allows to evaluate the inhibition or induction potential of a study drug for different enzymes or transporters in one study. Now, there are some criteria to be considered before conducting these studies. We need to use a validated cocktail to ensure that there is no interaction between the substrates included in the cocktail. And some of the examples of the validated cocktails which are used for conducting these studies are shown in this table on the right here. Now, the substrates in this cocktail are typically specific for the individual CYP or the transporters. Otherwise, the design of the cocktail DDI study is similar to a regular DDI study, which we have seen earlier, and the negative results from these cocktail DDI studies would eliminate any further DDI evaluations. Now, next, let us look at the mass balance studies. A mass balance study, also called as an ADME study, is recommended for nearly all drugs in the development. Now, the objective of a mass balance study is to understand the full clearance mechanism of the drug and its metabolites in the humans. This study involves administering a radio-labeled drug molecule, and the investigational drug is followed through the ADME process to track its absorption in the body, to evaluate the distribution in the plasma and the whole blood, to identify the metabolites of the drug, and to determine the relative exposure of the parent and the metabolites. Typically, these studies are conducted early in the drug development or at least conducted prior to the phase 3 studies. In some instances, they may also be conducted during the phase 2 proof of concept studies. Now, what we are hoping to learn from these mass balance study is to determine the overall pathways of metabolism and excretion for the investigational drug, finding out if there are any metabolites in the circulation, and then to determine the abundance of that metabolite relative to the parent parent drug or the total drug related exposure. Now, these mass balance studies are non-randomized, usually open label studies in healthy volunteers. Typically, a single dose is used, and at single dose clinical dose is used, and the common label use is C14. The study duration and the sample collection for these studies aims to maximize the recovery of the total radioactivity. And since per FDA guidance, these studies should aim to recover at least 90% or more of the radioactivity in urine and feces. In these studies, the concentration of the parent drug and the metabolites are measured, and the amount of radioactivity is determined in the plasma, urine, and feces. Since these mass balance studies provide valuable information regarding to the elimination of the drug and metabolites, these inform the clinical pharmacology section, specifically the excretion subheading under the elimination section in the drug label. Now, I'll touch on the hepatic impairment study, but before looking into the study considerations, let us look at a little background to know why these studies could be necessary. Now, we are aware that liver is a major organ involved in the clearance of many drugs via metabolic pathways or biliary excretion. Not only that, but liver also produces albumin, which could bind the drugs, and albumin binding plays a major role in clearance and distribution of the drugs. And hence, in the presence of liver failure or liver disease, which can either be acute or chronic, there would be reduced clearance of the drugs, or there would be less protein binding, leading to changes in the distribution and clearance. This could potentially impact the safety and efficacy of the administered drugs, and hence in liver disease, there could be a need to adjust the dose in order to mitigate these changes. Now, the objective of these hepatic impairment study is to assess the effect of hepatic impairment on the pharmacokinetics of the drugs and its metabolite, and if possible, to relate the specific measures of hepatic function or the group of function to a relevant PK measure. Now, hepatic impairment is classified based upon the clinical and the lab criteria as either mild, moderate, or severe hepatic impairment. Now, these studies could be either single dose or multiple dose studies. Sometimes population PK approach could also be used to assess the impact of altered hepatic function, where hepatic impairment could be tested as a significant covariate on the pharmacokinetics if these hepatically impaired patients are included in phase 2 and phase 3 studies and enough PK data is collected. For study design, these hepatic impairment studies could either utilize a full study design or a reduced study design. A full study design could include all the three groups, that is mild, moderate, and severe hepatic impairment patients, and compare them to a control group. The guidance recommend that each group should include at least 6 patients, and any recommendations that come from this study would apply to the entire spectrum of hepatic impairment. In a reduced study design, there are only two cohorts, and these are moderate hepatic impaired patients and the control group. The guidance recommend that there should be at least 8 subjects per group, and any findings that come from the moderate category would apply to the mild hepatically impaired patients. But for labeling purposes, the dosing in the severe category would generally be contraindicated unless further assessments in severe hepatic impairment hepatically impaired patients are conducted. Now, here is a decision tree which shows when a hepatic impairment study might be required and the type of the study to be conducted. Now, in case if the drug has a wide therapeutic range with hepatic metabolism accounting for greater than 20% of the absorbed drug, or if the drug has a narrow therapeutic index, irrespective of the proportion that is metabolized, an hepatic impairment study may be important. As I mentioned before, it could be either a full study design or a reduced study design could be used. And based on that, based on the results from the study, this would inform any dose adjustments and labeling recommendations. Now, since the main objective of the hepatic impairment study is to develop dosing recommendations so that the patients and the practitioners can alter the dose and dosing interval appropriately in presence of hepatic impairment, according to the FDA guidance, in order to recommend the dose adjustment in label for hepatically impaired patients, the effect of hepatic impairment on PK should be greater than twofold. However, exposure response analysis should guide this decision for dose adjustment. Now, in order to determine the dose recommendation is required or not, a confidence interval approach is preferred rather than a significance test. The sections that are informed are obviously the clinical pharmacology section, and the clinical pharmacology section of the labeling should include all the data to show the effect of hepatic impairment on the PK of the drug. As shown in the figure on the top here, it shows all the information or the data that is included in the label in case of a positive hepatic impairment study. The data from this study could also inform the specific population section in the label, the warnings and precaution sections, and the dosage and administration section. If the hepatic impairment study is conducted and it shows that it does not alter the PK, then it is also important to point that out in the product label. Or if the hepatic impairment study is not conducted when in fact it was required, the label should indicate that a hepatic impairment the hepatic impairment was not studied as shown in the box on the lower right corner here. Now, speaking of organ impairment, next we have the renal impairment studies. For drugs undergoing renal elimination, impaired renal function can alter exposures impacting the efficacy and safety of the drug. The renal impairment can impact the renal filtration, secretion, or reabsorption of the drug. It could also alter the drug absorption or the plasma protein binding, or it is also known to influence the metabolism in the liver and the gut, since impaired renal function is also associated with decreased activities of several hepatic and gastrointestinal drug metabolizing enzymes. Now, for drugs with high renal clearance, even mild to moderate renal impairment can impact the AUC by a twofold or more. While for drugs with a very low renal clearance, even severe renal impairment may have no impact. So for the purposes of renal impairment study, the renal impairment is classified based on estimated glomerular filtration or creatinine clearance as either mild renal impairment, moderate renal impairment, or severe renal impairment. Now, the objective of these renal impairment study is to estimate the impact of renal impairment on the pharmacokinetics of the drugs and recommend the dosing adjustments if necessary. Now, the FDA guidance recommends the use of estimated glomerular filtration rate assessment to determine the renal function for the purposes of the renal impairment studies. Now, these studies could be either single or multiple dose studies. Similar to hepatic impairment studies, the population PK approach may be used to assess the impact of altered renal impairment on the pharmacokinetics. Now, these studies could also be either full design study or a reduced design study. With full design studies, the guidance recommends using at least 6 to 8 subjects per group, and these compare the PK of the drug with varying degrees of renal impairment to the control group. Whereas the reduced design may be useful for the drugs which are non-renally cleared, but which are intended to be used in the renally impaired patients. Now, unlike the hepatic impairment study, these reduced renal impairment study compares the severe renal impairment patients to the control group. And usually these are considered as the worst case scenario. Under some circumstances, they may also warrant a full renal impairment study. Again, here is a decision tree which shows when a renal impairment study might be required and the type of the study to be conducted. Now, in case when the drug is intended to be administered in a renally impaired patients, a renal impairment study is recommended. If it is a non-renally cleared drug, then a reduced study design may be used. Whereas for a renally cleared drug, there is an option to use a reduced study design or a full standalone study design. The positive results from such study would require appropriate dose adjustments if clinically significant and appropriate labeling recommendations. Now, dosage recommendations in the renally impaired patients should be based upon the overall understanding of the relationship between the renal function, the drug exposure, and the exposure response relationship. For drugs with wide therapeutic range, the change in the drug's PK based on the renal function might not result in dosage adjustments for patients with renal impairment. Now, there are a number of approaches which could be used for dosage selections for patients with renal impairment. One is PK simulations could be conducted to project the systemic exposures for renally impaired patients that fall within the 5th and the 95th percentiles for those in the reference group or the control group. Or the second approach is to establish the no effect boundaries, which is an interval within which the change in the systemic exposure is not significant enough to warrant any clinical action. Now, these renal impairment study support the labeling recommendations in the following sections. It informs the ClinPharm sections, specifically the pharmacokinetic subsections, which includes the renal elimination of the drug and its metabolized, any information on the PK/PD changes relative to the renal function. The specific population section could also include any dosing adjustments which are required specifically for the renally impaired patients, or if any specific monitoring recommendations needed would be included under this section. The warnings and precautions sections, contraindication sections, and the dosage and administration sections would also be informed based upon the results from these studies. Now, additional studies to support the filing and labeling are evaluation of the QT interval prolongation, the pediatric study, and the lactation study. For the purposes of today's talk, I'll just be discussing the study to assess the QT interval prolongation. Now, why is it essential to evaluate the QT interval prolongation potential of an investigational drug? When there is a delay in the cardiac repolarization, which is measured as the prolongation of the QT interval on the ECG, there is a potential risk of torsades de pointe or TDP, which could result in sudden cardiac death. The ICH E14 guidelines recommends that all new drug applications assess the potential repolarization delays after the administration of the investigational drug by assessing the effect of the drug on the QT interval. Now, just a point to note here is that since the QT interval is strongly influenced by heart rate, the QT interval is often expressed as the corrected value, or the QTC, that accounts for the heart rate. Now, evaluation of the QT prolongation could be done by conducting a thorough QT study. These are randomized, double-blind, placebo-controlled crossover studies. Sometimes parallel studies might also be done. These could be done with a single dose or multiple doses. These are conducted usually in normal healthy volunteers, and typical treatment arms include a placebo arm, a positive control, which is included in the study to demonstrate the study's ability to detect the effect on the QT interval. Usually moxifloxacin, which has a mild QT prolongation effect is a commonly used positive control in these studies. It includes a therapeutic dose of the investigational drug, and it also includes an arm with which includes a supratherapeutic dose of the drug in order to represent the worst case scenario, such as which might happen with the drug-drug interaction or food-drug interactions, or in case of organ impairment patients. And typically this dose is the one which shows 3 to 5 times the clinically relevant exposures. Now, the concentration QT analysis could be conducted to obtain a TQT study waiver. Concentration response analysis is typically conducted by pooling all the available data across all the doses, typically from the first in human studies. This requires the availability of robust ECG data from these early phase studies. The CQT analysis evaluates the QT interval response at greater than 2 times the high clinical exposure. When sufficient multiples of high clinical exposures are not included in the early studies, or a conventional TQT study is not possible, an integrated non-clinical and clinical QTC risk assessment could be performed. Now let us look at the implications of QT prolongation as determined by a TQT study. Since the objective of the TQT study is to exclude any QTC effect by the investigational drug, a positive TQT study would mean that an effect of the drug on the QT prolongation should be extensively studied during the late phase studies. In some cases, that could also result in the non-approval of the investigational drug. Last year, FDA issued a guidance regarding the QTC information to be included in the drug labeling. Now, evaluation of the QT prolongation could inform the following sections in the drug labeling. It could inform the clinical pharmacology sections. The relevant information on the effects of the drug on the QTC interval described under the cardiac electrophysiology heading in the pharmacodynamics subsection. It could also include any dose or exposure response relationship for the drug. The drug-drug interaction section uh, could inform if there are any clinically significant DDIs that increase the risk of QT prolongation. Um, Warnings and precautions sections, contraindication sections. In case of QT interval prolongation, when the result which results in the cardiac death or life-threatening adverse events, then the boxed warning sections would be included in the label. These studies would also inform the dose and administration section, the adverse event sections, and the patient counseling information section. So in summary, we have seen how these ClinPharm studies represent a critical component in the clinical development. These clinical pharmacology studies help generate valuable information that helps guide dosing regimens or recommendations to obtain optimum benefit risk profile for the drug. These studies help understand the safety and efficacy of the drugs in a specific populations, such as organ impairment, patients with organ impairments. These studies also help guide the study design considerations for the late phase studies. And thus, due to all of this valuable information, these studies help support the NDA filing and labeling claims. This concludes my presentation for today. Thank you everyone for your attention.